Hello everyone, and today in this topic we'll learn about the protoplast culture and the somatic hybridization. We'll learn today the basics of the protoplast culture, what is exactly protoplast culture, then what are the historical developments related to the protoplast culture and the importance of the protoplast culture. Then we'll also learn about the isolation of the protoplast via the mechanical method and the enzymatic method. So let's begin now. Now, coming to the protoplast culture, what is exactly protoplast culture? Many a times in plant tissue culture, we go through the concepts of the protoplast culture, but what it is exactly? So, protoplast culture are the naked plant cells without the cell wall. So, protoplasts are the naked plant cells without the cell wall, and they possess plasma membrane and all other cellular components. So, this is protoplast. So, protoplast culture. So, culturing of protoplast is done. But what is exactly protoplast? Protoplast are the naked plant cells without the cell wall. They, but they possess the plasma membrane and all other cellular components. Now they represent the functional plant cells, but for the lack of the barrier cell wall. So they represent the functional plant cells. And the protoplast of different species can be fused to generate a hybrid, and this process is called as somatic hybridization. Now, protoplast culture is the culturing of protoplast is done, but the different protoplast of different species can be fused and then it will generate a hybrid. And this hybrid is nothing but it is called as somatic hybridization, that the protoplast of different species are fused together. Then there is a phenomenon called as cybridization. Now, what is exactly cybridization? Cybridization is a phenomenon of fusion of a normal protoplast with an enucleated. Now, enucleated is what? Without nucleus. So, the phenomenon of fusion of a normal protoplast with an enucleated protoplast that results in the formation of a cybrid or cytoplast. That is, the cytoplast means the cytoplasmic hybrids. So, that is cybridization. Cybridization, it is the fusion of a normal protoplast with the without nucleus protoplast. So one is the normal protoplast with nucleus and the other is without nucleus. That fusion, it results into the formation of a cybrid or a cytoplast, that is cytoplasmic hybrid. That is called as cybridization. So in our protoplast culture and somatic hybridization, these three concepts are very important. That what is exactly protoplast culture, then what is somatic hybridization, and what is hybridization. So these are the important concepts. Now coming to the historical developments. Now how this historical development took place and who did that? So the term protoplast was first introduced in 1880 by Hanstein. Hanstein was the person who introduced the term protoplast in 1880. So the first isolation of protoplast was achieved by Klucker in 1892 employing a mechanical method. Now there are two methods in the protoplast culture. One is the mechanical method and one is the enzymatic method. So the first isolation of protoplast, which was done by the mechanical method by the Klucker in 1892. Then a real beginning in the protoplast research was made in 1960 by Cocking, which used the enzymatic method for the removal of cell wall. So for the removal of cell wall, the enzymatic method was used, which was done by Cocking. And Recabe and his associates in 1971 achieved the regeneration of whole tobacco plant from protoplast. So Recabe was the one who achieved the regeneration of whole tobacco plant from protoplast in 1971, and then after that rapid progress occurred after 1980 in protoplast fusion to improve the plant genetic material and also to develop the transgenic plants. Now, for the development of transgenic plants and to improve the plant genetic material, further development took place. So this is about a bit about the historical development in protoplast culture. Now, coming to the importance of protoplasts and their cultures. Now, the isolation, culture, and fusion of protoplast is a fascinating field in plant research. We know that. So, plant protoplast isolation and their cultures provide millions of single cells that are comparable to the microbial cells for a variety of studies. Now, they have wide range of applications. Why they are important? Why the importance of protoplast culture is there? Because of the applications, variety of application it provides. So the first one is the protoplast in culture can be regenerated into a whole plant. So the protoplast in culture can be regenerated into a whole plant. Hybrids can be developed from the protoplast fusion. We learned that what is how the protoplast fusion is done in detail. So hybrids can be developed from the protoplast fusion. Then it is easy to perform the single cell cloning with protoplast, and the genetic transformation can be achieved through the genetic engineering of protoplast DNA. Then the protoblasts are excellent materials for the ultrastructural studies. Now, in order to do the ultrastructural studies, the protoplasts are the excellent materials. Isolation of cell organelles and the chromosomes is easy from the protoplast, and protoplasts are useful for the membrane studies. That is, for transport and uptake studies, protoplasts are very useful, and also for the isolation of nutrients from the protoplast culture is easy. So this is the reason why that it has got a variety of applications, and therefore there is importance of protoplast and their culture. This is the importance of protoplast and their culture. Coming to the isolation of protoplast. As I said before, that isolation of protoplast, it has got the two method. One is the mechanical method, second is the enzymatic method. So in the historical developments, we learned that in 1892 first the mechanical method was developed, and after that the in the 1971 the after that in 1892 was the time period where the mechanical method was developed, and 1960 was the period where the enzymatic method was developed. So what is this mechanical method and enzymatic method? This we'll learn in more detail. Now, the mechanical method, protoplast isolation is done, and it is a crude and the tedious procedure. So it results in the isolation of very small number of protoplasts. But then what are the stages that are involved in the mechanical method? Now from the figure you see, you will be able to observe that a small piece of epidermis from a plant is selected first for the mechanical method, and then the cells are subjected to plasmolysis. So this causes the protoplast to shrink away from the cell wall. So second step you can see first is the epidermis, small epidermis is taken from a plant, and then the plasmolysis take plasmolysed cell. So plasmolysis takes place. It causes the protoplast to shrink away from the cell wall. The third step is that tissue is dissected to release the protoplast. So you can see from the figure that the tissue is dissected to release the protoplast. So the protoplast is released. So the mechanical method for protoplast isolation is no more in use because it has got some limitations. Although the method is quite easy, but it is a very tedious process, and it is laborious also. So it requires a more amount of labors as well as it is tedious process. Then it is restricted to certain tissue cells. It is restricted to certain tissues with the vacuolated cells, and the yield of protoplast and their viability is also low. So the mechanical method it has got some limitations. And mechanical methods of the cell wall degrading enzymes of enzymatic method cause the deleterious effects to the protoplast. Then the workers prefer the mechanical method. So this is about the mechanical method, that how it is carried out. Once again, I'll clear this concept that first the small piece of epidermis is taken from the plant, Then the plasmolysis takes place, where a small plasmolysed cell, it is what it is done, the cells are subjected to plasmolysis, and then the tissue is dissected from the cell, which is the plasmolysed cell, and then the protoplast is allowed to released. So this is the mechanical method for the isolation of protoplast. We know that from isolation of protoplast is nothing but the protoplast is removed from that plant. So this is the isolation of protoplast is carried out by the mechanical method. Now coming to the enzymatic method. Now enzymatic method, it is widely used technique for the isolation of protoplast, and it has got the advantages that it gives a good yield of the viable cells, and there is no damage that is caused to the protoplast. So because of these factors, the enzymatic method is widely used. Now, the sources of protoplast, it can be isolated from a wide variety of tissues and organs that may include the leaves, roots, shoot, apices, fruits, embryos, and microspores. From these are all the sources from where the protoplast can be isolated. So, the mesophyll tissue of fully expanded leaves of young plants or the new shoots are most frequently used ones. In addition, the callus and suspension cultures, they also serve the good sources of protoplast isolation. So the sources of the protoplast, we know that mesophyll tissue of fully expanded leaves or the new shoots or the callus and suspension culture are the uh, good sources for the protoplast isolation. But then there are also certain enzymes that are required for the isolation of protoplast. So the enzymes that can digest the cell walls are required for the protoplast isolation. Chemically, the plant cell is mainly composed of cellulose and hemicellulose and pectin, which are degraded by the enzyme cellulase. We know that cellulose, it is degraded by the enzyme cellulase, then the hemicellulose by hemicellulase, and the pectin by pectinase. So these are few of the enzymes that are used for protoplast isolation. The enzymes for protoplast isolation are commercially available, and the enzymes are used at the pH of 4.5 to 6 temperature, and the temperature that is used is 25 to 30 degrees Celsius. So pH which is used is 4.5 to 6, and temperature is 25 to 30 degrees Celsius with a wide variation in incubation period. It may range, the incubation period may range from the half an hour to 20 hours. So these are the enzymes that are used, that is cellulase, then hemicellulase and pectinase. So first we learnt about the sources that are used, then second we learnt about the enzymes that are used, because it is enzymatic method. So enzymes, they are necessary, and it is necessary for the enzymatic method to use the enzymes. Now, there are two steps or two approaches that are involved in this enzymatic method. One is the two-step method and the other is the one-step method. Now, this two-step method, it is also called as sequential method. So, it is two-step, and the one-step method, it is also called as simultaneous method. Mechanical method, we already learned, it is only given in the return format, that how the mechanical method is carried out, and then what are the limitations of the mechanical method. Now we learn in detail about the major steps that are involved in the protoplast isolation. So the two-step method, what it is done is the tissue is first treated with pectinase enzyme to separate the cells by degrading the middle lamella. So first the tissue is treated with the pectinase enzyme to separate the cells by degrading the middle lamella, and these free cells are then exposed to the cellulase to release the protoplast. Now, when the free cells are exposed to cellulase to release the protoplast, what pectinase does is that pectinase breaks up the cell aggregates into the individual cells, while what cellulase does is it removes the cell wall proper. So the two-step method, why it is called, that there are two enzymes that are used. First is the pectinase and the other one is the cellulase. So what pectinase does is that it breaks up the cell aggregates into the individual cells, while the cellulase, what it does is it removes the cell wall proper. So this is the two-step method. Now coming to the one-step method, or it is also called as the simultaneous method. Now what it is done in this method is it is preferred method of for the protoplast isolation, and it involves the simultaneous use of both the enzyme, that is the um, cellulase enzyme and also the pectinase enzyme. So one step method, it is also where the uh, simultaneous use of the both the enzymes are done. So two-step method, first it is treated with pectinase and then it is treated with cellulase, while in one-step method both the enzymes are used simultaneously. So pectinase and cellulase, both of them are used simultaneously in the simultaneous or the one-step method. Now, isolation of protoplast from leaves. Now, leaves are the most commonly used for protoplast isolation since it is possible to isolate the uniform cells in large number. So, first what it is done, that the sterilization of leaves is done. So from the figure you can see that from young plant leaf is taken and then the leaf sterilization is done. Then further the removal of the epidermal cell is done. So epidermal cell removal of epidermis from the cell layer is done. That is the second step. Then it is allowed with to treat with the enzyme. So epidermis it is allowed to treat with the enzyme. So from the figure you can get that first the young plant is taken, then the leaf sterilization is carried out, then the treatment epidermis removal is done from the cell layer, then the treatment is done with enzymes, and then the isolation of protoplast is carried out. So the isolation of protoplast, it is carried out from leaves because it is commonly used method. Now besides leaf, callus cultures and cell suspension cultures can also be used for the isolation of protoplast, but for this purpose the young and actively growing cells are preferred. So first that this is how the isolation of protoplast is done. Now the next step is involved, that is the purification of protoplast. Now when first we are treating the epidermis with the enzymes, that is the isolate, and then the isolation of protoplast is done. So the purification of protoplast, it is also necessary to purify the protoplast. So the enzyme that is digested plant cells besides protoplast contains the undigested cells. Now the broken protoplast and undigested tissues, the cell clumps and the undigested tissues can be removed by filtration. Now, after that, it is followed by the centrifugation and the washing of protoplast. Now, you can see from the figure that when it is treated with the enzyme, cell is treated with the enzyme, and then the cell wall dilation takes place and the release of protoplast is taking place. And then further what happens is the protoplast is there, and then there are also cell debris that are present. So centrifugation is carried out and the protoplast is released, a protoplast is taken and cell debris are not taken. After that, the washing of protoplast takes place. So the protoplast gets isolated, right? So this is the isolation of protoplast. Now, this is the isolation of protoplast which is done. After that, what happens is after centrifugation, the protoplast are recovered above percol. So above percol, the protoplast is uh, is, uh, protoplast are recovered. Then the viability of protoplast. Now it is necessary to ensure that the isolated protoplast are healthy and viable, so they are capable of undergoing the sustained cell division and regenerations. Now for that several methods are carried out for the viability of protoplast. One is the fluorescent diacetate staining method that is carried out. So in this, the dye accumulates inside the viable protoplasts, which can be detected by the fluorescence microscopy technique. Then the other is the phenylephrine stain. Is the it is selectively taken up by dead protoplasts. The dead protoplasts turn red, while the viable cells remain unsustained. So this is the phenylephrine stain that is carried out. The third one is the exclusion of Evans blue dye by intact membranes. Fourth method is the measurement of cell wall formation. Now for that the CFW, that is chalcofluor white stain, binds to the newly formed cell walls and they emit the fluorescence. The fifth one is the oxygen uptake by protoplasts that can be measured by oxygen electrode. And the sixth method is the photosynthetic activity of protoplast. Seventh one is the ability of protoplast to undergo the continuous mitotic division. Now this is the direct measure that is carried out. So this is the uh, isolated protoplaster. Then they are done. Then after that the plating of protoplast is carried out. Further the cell wall regeneration takes place. Then the clump of cells are formed. After the clump of cells the callus tissue is formed. Then the differentiation of callus tissue takes place, and then the plantlet are derived from the protoplast, and then further again it grows into a young plant. So these are the isolation of protoplast, culture and regeneration of plants. These are the steps that are involved. Once again, we'll study this, that first a young plant is taken, then the leaf sterilization takes place, then the epidermis is removed from the cell layer. After that, the enzymes are introduced to the epidermis. Then after the cells in enzyme mixture, after that what happens is the cell wall digestion and release of protoplast takes place. Then with the help of centrifugation, the centrifugation is carried out and the protoplast are separated. Then the washing of protoplast is done. Now the isolated protoplast, they are taken and then their plating of protoplast is carried out. After that, the cell wall regeneration takes place. Then the clumps of cells are formed. Further, the callus tissue formation takes place, and then the differentiation of callus tissue takes place, which then forms the plantlet that is derived from the protoplast, and then it forms into the young plant. So this is the enzymatic method of the protoplast. Then the purification of protoplast, we studied that how what are the stains that are required for the purification of the purification of protoplast, why it is important. We studied about that. Then we also studied about the viability of protoplast, that for the viability of protoplast which are the different stains that are used and how the methods are carried out. So isolation of protoplast from leaves, we already studied that what are the steps that are involved. Then the purification of protoplast, how it is done, we also already studied that. The enzymes digested plant cells, protoplast contain the undigested tissues, cells and protoplast. Then the cell clumps and the undigested tissues can be removed by filtration, centrifugation and washing of protoplast takes place, and then the protoplast are removed from above per call. So this is the purification of protoplast. Now from the figure also you can see that how the purification of protoplast is done. We already learned about the viability of protoplast. So these are the stains that are used in viability of protoplast. One is the fluorescent diacetate staining. Second is the phenyl stain. Third is the Evans blue dye. Fourth one is the measurement of cell wall formation. Fifth is the oxygen uptake. Sixth one is the photosynthetic activity, and seventh one is the mitotic division. So this is about the viability of protoplast. So today we learned about the enzymatic method, then the mechanical method, some of the historical development, then the importance of the protoplast culture. In the coming lessons, we'll learn more about the culture of the protoplast, then the protoplast fusion, then the applications of the stomatic hybridization, how the stomatic hybridization is carried out, then what are the limitations to the stomatic hybridization. So stay tuned for more lessons to come. And if you like the video, don't forget to click on the like button below. Also subscribe for more video updates, and also don't forget to comment because your comment matters a lot. So don't forget to comment below, and thank you for watching.